Okay, so this is the urea cycle. So here you're going to have nitrogen is going to be transanimated to create ammonia and intermediates of the Krebs cycle. So a lot of the nitrogens that we're going to be getting, they're going to be coming from amino acids, and then also from the breakdown of nucleic acids. Okay, so ammonia is going to be basically processed in the urea cycle, and then it's going to be, you're going to be producing urea that is going to be eliminated to the kidneys. So anything that isn't going to be utilized is going to be made into urea, and it's going to be gotten rid of by the kidneys. Okay, so in the mitochondria, so we have, at the beginning, we have carbon dioxide. Carbon dioxide is going to be changed into bicarbonate. And then you're going to get ammonia, I mean, not ammonia, ammonium, the ammonium ion, because some of the ammonia is going to be converted to ammonia. Some of the ammonia is going to be converted to ammonium. And so you're going to have, it's going to take that ATP, use that energy, the enzyme carbonyl phosphosynthase is going to actually utilize the ATP and use that inorganic phosphate. And then you're going to get the production of carbonyl phosphate. Carbonyl phosphate is going to then lose its phosphate, and it's going to combine with ornidate. And so the ornidate and the carbonyl phosphate is going to be utilized to sit there and make L-citrulline. L-citrulline, it's a modified amino acid that isn't part of your normal, part of the normal amino acids that you use for like muscle or protein polymerization. So it's just utilized in this cycle only, and it's basically used as kind of like a carrier or a transitional protein, an intermediate. So the L-citrulline gets processed and modified to argiosuccinate. And the way it does that is that you have the L-citrulline that acts as like as a base. This arginosuccinate synthase combines the L-citrulline and it combines L-aspartate together, and then that L-aspartate will end up being modified to sit there and make the arginosuccinate. So an L-aspartate is one of your amino acids that are found in muscles. No, let me see if I can get this. Okay, so we're down here, right? So we have arginosuccinate. The arginosuccinate is then it's going to be cut essentially, and you're going to get fumarate, and you're going to get L-arginine, right? So we essentially are getting the production of that arginine from the combination of the citrulline and the aspartate. So then that L-arginine, which is one of the amino acids, is then going to be further processed, and you're going to end up getting urea and ornithine, to go back to the beginning of the cycle. And then the ornithine is reused and you reuse it through that whole process again. Okay.
Mm-hmm.
So again, so if needed, if we need more arginine, we're able to sit there and get it from the production of this. If we have excess of this, it's going to continue and just be broken down into urea. This guy's going to go through that process.
Okay.
Okay. Okay, so the way that we use energy from amino acids can basically be broken down. The cool thing about amino acids is that they can interact at various points. Right, so we talked about the 3 stages of energy production. I said we have glycosylation, we have Krebs cycle, and then we have the ETC, electron transport chain, or we call it the oxidative phosphorylation. So those are your 3 cycles. So the cool thing is that amino acids could actually go in. They could go into the glycosylation component of the cycle. Hold on. Too many things. So they can go in through the glycosylation component of glycosylation, the glycolysis stage of the cycle. In the glycolysis stage of the cycle, you're going to be inputting towards the last part of glycolysis. So when you get that pyruvate molecule, so you have the glycogenic amino acids. They're able to sit there and enter at this point to start at the production of pyruvate, and then they just go through, and then they make this acetyl CoA, and then we know that that's when it enters. It's entering the mitochondrial matrix. So it enters at the mitochondrial matrix at this point. So that's 1. So if we look at that, that's 5 amino acids. We have adenine, cysteine, glycine, serine, and threonine. Right? And if you kind of look at the characteristics of those amino acids, all of those amino acids, they're kind of water friendly, right? So they're able to sit there and be out in solution. They're not hydrophobic like. So all 5 of these are sort of that way, right?
Mm-hmm.
Here we have the ketogenic amino acids. So these ketogenic amino acids, these guys are going to be basically using the ketone oxidation, and that should be an N, not an S. So ketone oxidation for the entry at the acetyl CoA component. Okay, so the characteristics about these guys, they're typically hydrophobic. So they're often using the hydrophobic structure of it. Okay, so that's 1, 2, 3, 4, 5, 6. We have phenylalanine, tyrosine, leucine, lysine, tryptophan, and isoleucine. Okay, so we have other amino acids that kind of their structure are very similar to the keto glutarate. And so since they have something similar to that, they're going to sit there and be utilized to enter at that cycle where ketoglutarate is going to be generated. So then you have your, again, we have arginine, we have histidine, we have glutamine, we have glutamate or glutamic acid, and we have proline, okay? Now, these guys too, they're also gonna be hydrophilic. They're gonna be able to interact. They're gonna have a charge associated with them. And so we have these 5 amino acids as well. So if you remember correctly, we have a total of 20 amino acids that are utilized for making of protein, okay? And right now we've basically accounted for, what is that, 15, 16 of them?
Yep.
Okay, so down here we have the isoleucine, methionine, and valine. All of those are hydrophobic, and they're going to enter at the succinyl-CoA part of the Krebs cycle. Now, this guy has already been accounted for. This one has accounted for here. The other two, this is the only part where they're going to enter that. Okay.
Okay.
So we have, now we're up to what, 17? That's right. 11. Yeah, 18. Right, 18. Okay. So then at the fumarate part of the cycle, you have phenylalanine and tyrosine, which we've already accounted for earlier on. And then at the oxoacetate, we have aspartic acid and asparagine. Okay, so that's going to be your other two. So if we count right, that should be a total of 20. So we have 10 plus 6 plus 2 plus 2. So that should be 20. So all 20 amino acids. And the cool thing is that they're entering at different points. So if one point is kind of get screwed up, you always have another point of entry. And so you're always able to sit there and make energy. You know, you may be making a lot less since you're not able to use all of the amino acids, but you're still able to make energy to sit there and sustain yourself.
All left over. That's good.
Okay, so this here is just the big picture of metabolism, right? So we know that metabolism is composed of metabolism and anabolism. And so this is the catabolic pathways represented by the arrows going, the blue arrows. Let's see, by these blue arrows, and I guess the red arrows here. And so that is going to be your catabolic pathways. And then you have your anabolic pathways, which are going to be acting reverse to that. And so it's kind of cool. You have things that are going in both directions. So you have a sense of an equilibrium, balance that is presented by the metabolic pathways. Okay. Let's see. So this here is the absorptive state. So when the body digests food, it's going to basically need to absorb nutrients. And so this is sort of like the process that we'll have to go through to make that happen, right? So initially, the first thing that we do, we get in our nutrients, we chew it all up and everything like that. We digest our nutrients, and so we end up with the bare bones, right? We have our glucose, we have our lipids, and we have our amino acids, okay? So that is going to be going in our bloodstream, and then so it's going to be entering our bloodstream via our intestines. So then our blood sugar, as a result of that, is going to rise up. And so we're going to have that monitoring system that recognizes that our blood sugar has risen up. As a result of that, our pancreas is going to start secreting insulin. And we know that insulin kind of is responsible for regulating that blood sugar so that our blood sugar doesn't get too high, right? So then after the insulin is released by the pancreas, that insulin is going to have the cells to start taking up glucose. And then so that we can actually reduce the sugar in the bloodstream. So here in the liver cells, they're going to actually start taking up the glucose, right? So they're going to have the receptor that says, hey, an insulin receptor that tells it, hey, you need to start uptaking some of that glucose. And so what's going to happen is that your liver cells are going to sit there and take up the glucose. It's going to take all that excess sugar and it's going to store it as glycogen. And it's going to be used for storage for later on when we need to sit there and make more energy. Okay, so also the amino acids, they're going to be converted into ketone bodies. Okay, so that they can be later converted to acetyl-CoA when we need to sit there and convert them to acetyl-CoA. The muscle cells are going to sit there and they're going to be told, hey, you need to absorb some of that glucose as well. So they're going to take up that glucose. Again, they're going to be storing it as glycogen. And then the amino acids are going to be used for synthesis of proteins. So you're going to be making actin and myosin, and those are going to be used to build your muscle fibers. The adipose cells, well, they're going to sit there and say, okay, so the insulin is going to get there. It's going to cause the adipose cells to sit there and start storing lipids. So all the lipid structures that are left are going to sit there and be stored in the adipose cell itself. And so then that's just going to act as a reserve for fats that we may need to use for energy later on, or for lipolysis. Okay, so post-absorptive state. So that's basically the state after we've absorbed. So we've already absorbed what we wanted to absorb. Now our sugar levels and everything are really low. We don't have any sugar in our system because we just didn't eat. It's been a while since we ate. And so our body kicks in immediately. So it kind of recognizes that, hey, I'm a little bit hungry. So the first thing that it does, it's going to start sending out your glucagon. So then your glucagon, because your blood sugar is so low, you're going to sit there and get the release of your glucagon. Glucagon is going to be released into the system. And then glucagon is going to go to the liver cells. It's going to sit there and start stimulate the liver cells to sit there and start releasing glucose. So the way that it's going to start releasing glucose is by glucogenolysis. And in that case, you're basically going to be cutting up glycogen molecules so that you're just getting those glucose molecules. And then you're also going to get ketone oxidation, which is going to sit there and it's going to sit there and break down your ketones, or it's going to utilize those ketone bodies that we had stored earlier, and then also utilize some of the amino acids and start catabolizing some of those amino acids. Okay, and you're also going to get release of lipid structures and glucose, so that your body can actually use it to start making ATP. So then your blood, all of that's going to be released and led into the bloodstream and stuff like that. So you're starting to get a little more production of glucose, your lipids, and your amino acids. Okay, so in this case, your muscle cells is going to, at the same time that this is happening, your muscle cells are also being stimulated by the glucagon. The glucagon is going to sit there and cause, again, oxygen to be released. Glucose to be released. And that release of glucose is going to be coming again from the glycogen. Glycogen is going to be broken down. And then that glycogen is going to be broken down to glucose 6-phosphate. And then glucose 6-phosphate is then going to be broken down to just glucose. So the amino acids, they're going to come from the actin protein, actin and myosin. So actin and myosin is going to be catabolized and going to be nicely cut up. And actually, it's going to be delivered and utilized by the liver, okay, to make our sugar molecules. Okay, and so the last thing that's going to happen is that glucagon is going to stimulate the adipose tissue to release some of the stored lipids. And so then it's going to release the stored lipids. And at that point, those lipids are going to be kind of delivered to the liver, and the liver is going to start processing the lipid structures to sit there and make more ATP is needed. Okay, you got how it works, and see how, you know, this, you have those three types of cells that are going to be utilized through metabolism.
Yep.
Okay. The hypothalamus. The hypothalamus is going to be responsible for controlling thermoregulation. So we kind of talked about thermoregulation before, and we talked about how we get constriction, and so your blood flow is going to be kind of closer towards the inner parts rather than the outer parts when it gets cold, and then it gets towards the outer parts when it's hot, so we can cool off the surface, our surface, to try to keep us kind of cool. So the way it actually works is that, you know, of course, first it starts off with our body temperature. Our body temperature is low, then our temperature sensor is going to cause the hypothalamus to be stimulated. So we're going to send a signal to the hypothalamus. Hey, you need to control that heat. Control the temperature. It is freezing, right? And so the hypothalamus is going to say, hey, we need to produce some heat. So it's going to send the signals to produce heat. And then what happens is the superficial arteries are going to be constricted, and so they're going to actually reduce heat loss. So in other words, blood isn't going to be flowing to the top component where you're actually going to be losing some of that heat, right? It's actually going to be done closer, so you get that constriction, so that your blood is going to be flowing closer to the inside. And then overall, your blood flow to like your digestive system is also going to decrease. Okay, so you're going to lose access to the surface, the closer to the skin surface, so you're not losing heat that way. So heat isn't being transferred to the outside in this case, because you're freezing, right? You want to try to keep that heat in. You're also going to start shivering, right? You feel really cold and everything like that. The reason why we shiver, why do we shiver? I think you even mentioned this the last time.
Make yourself warmer.
To make yourself warmer, right? So during this process, shivering, you're going to start to increase cellular respiration, aerobic respiration. Although you did not take my chemistry class yet, I talked about this a lot. C6H12O6. Plus O2 yields, sorry, I'm in the middle of equilibrium, so everything is reversible right now. Carbon dioxide plus H2O plus ATP plus heat. Okay, so your muscles are going to start doing this. They're going to go through the process where they're going to start utilizing that glycogen, use that glycogen to sit there and make your glucose. That glucose is then going to be processed for cellular respiration. You're going to get ATP, energy, and you're going to get heat generated. Okay. And finally, your thyroid is going to kick in. I should say finally, it all happens at the same time. Your thyroid is going to kick in, and then you're going to go through the process. Excuse me. Of having cells to increase metabolism as a whole. So your thyroid is going to cause that metabolism to produce more heat. Okay. So your body temperature is going to increase, and then now you're warm. Okay. Now what happens when your body is hot? It's a hot day. It's 150 degrees outside, right? Your temperature receptors going to recognize that. They're going to the thymus. The thymus is going to release. It's going to initiate a heat release mechanism. So then it's kind of opening up the floodgates in this case. So it's going to open up your superficial arteries, so they're going to dilate. And because they're dilating, they're going to actually be able to sit there and help the system cool off. So you're allowing heat to be released to the outer surface. So you start to sweat, you're steaming up and all that stuff, right? So blood is going to get diverted away from the digestive system. And so you're also going to start sweating. And then the thyroid is going to decrease the metabolic heat production. So in this case, you're not going to be producing a lot of ATP or anything like that. You basically are not going to want to do that because in this case, you're going to be, warm, right? You don't need any more heat. And have you ever noticed, like on really, really hot days, your appetite, are you hungrier or are you thinking about food on really, really hot days and you're out there?
Like about ice cream.
You say you're thinking about ice cream?
Yeah.
Is that to cool off or is it for the taste of ice cream?
Just cool off.
Yeah.
Would you feel more hungry in the heat?
What was that?
Would you feel more hungry in the heat?
No, typically your appetite is suppressed. You don't want to eat as much.
You don't want to use too much energy, right? You want to cool off.
Exactly. Well, you don't want to use so much energy. Part of that is because you're already hot, right? So you already have that heat component. And so here you have an increase in heat. So it's going to actually be driving the equation in the other direction. So you want to start storing that stuff. So if we were able to sit there and make glucose from CO2, which we're not able to do, we'd want to drive that reaction. But in this case, what's happening to our glucose is pretty much being stored because we don't want it right now. We don't want to use it because we don't. We're trying to cool off. Okay. So at the end of the day, metabolism has to be controlled. And part of that control is a result of us, how we eat, our nutrition. And let's see. If we think about our nutrition, we get a lot of our nutritious elements from our food. We get the four major groups. We get our lipids. We get our lipids. We get our protein. We get our complex sugars from food sources. And we also get other things as well. So we get some minerals, and we get the abundant minerals, so stuff like sodium, and we get the stuff that are trace elements. We get our vitamins from various sources, like for instance, vitamin D. We actually we use sunlight to actually synthesize vitamin D. So.
Yep.
So nutrition plays a huge part in things as well too. There's something else I want to say about that. And so if we think of our vitamins, we have fat-soluble vitamins and we have water-soluble vitamins. And they're the ones that are hydrophobic. Those vitamins are going to be basically used in areas where we are going to need the hydrophobic. They're going to need to be absorbed or trafficked through a membrane. The ones that are hydrophilic, they're often going to have to be brought in by ion channels or coupling through channel or pores. Okay.
Mhm.
Let's see. And if we're not taking in these minerals and everything like that, that can lead to problems, right? So, like for instance, if we don't get enough potassium, well, we know that we need potassium for nerve function and muscle function. So it acts as an electrolyte for us. If we don't have enough potassium, we can sit there, it causes us to be weak, it causes us to be fatigued, causes cramping, it causes gastrointestinal problems, cardiac problems. If we don't get enough magnesium, we're agitated, we're anxious, we can't sleep. We often will get nauseated, vomiting. We have abnormal heartbeats. We have a low blood pressure. We're also going to have problems with our muscles. All right. So a lot of these minerals and vitamins play a huge role in our normal day-to-day metabolism. We need it, you know, and without it, it causes huge problems. Okay, so chapter 25, and that's the last chapter.
Stretch.
Okay, chapter 25, we're going to be talking about the, skipped everything, the urinary system. So this is basically a sewage plant. And so we basically know that our body needs to get rid of waste, right?
Mm-hmm.
And part of that way that we use urine is to get rid of those waste molecules, or those waste byproducts. Okay. So this is urine. So this is the color of urine. So if we're hydrated, our urine is really light in color. The more dehydrated that we get, our urine becomes darker in color. Okay. So the urine actually kind of serves as a good way of letting us know what's going on with our body, right?
Mm-hmm.
If our urine has a pinkish color to it, we often will know what's going on in that case.
Kidney stones.
What was that?
I only think of kidney stones if it comes out that color.
Yes. Well, the reason why kidney stones are causing that to happen, that pinkish color, is because you're getting cutting.
Yep.
Of probably somewhere in your urinary tract with the stone itself, and it's leading to blood. So typically, if we see red or pink in our urine, we know that there's some type of internal damage that is going on, right? Like you said, stones, right? In the case of, yeah. Okay. As we said, by it being brown or dark in color, we know that we're dehydrated, that we need to sit there and take in more water. You know, often my pee falls really deep in that dark, you know, because I know I don't get enough water. And so it often tells us a lot about what's going on with our diet as well, too, the fact that I'm constantly dehydrated. Other things, if the volume itself, how much pee are you peeing? So a normal person pees 1 to 2 liters a day. Actually, let me take one step before we talk about the volume. Fact. So we often pay attention to the various characteristics of, like the color. We also pay attention to the odor. So if the odor is off, so usually if your urine is in the normal range, it's gonna be odorless, or not too much odor unless it's been sitting around a long time.
Yeah.
Its pH is going to be anywhere from 4.5 to 8. So, but it could be, if it's really low, if your pH is low, you know that there's something wrong. If your pH is high, you know that there's something wrong. And so you should have very little to no blood cells in your urine. You should have little to no protein in your urine. So if you know that you have high protein in your urine, you know that there's something wrong. If you know that you have high amino acids in your urine, you know that there's something wrong, because all of those things should be filtered out and recycled by the body. And you should have no, if under normal circumstances, you should have no ketones, you should have no glucose, right? So if you have glucose or ketone, that also says that there's something.
It'll be diabetic, right?
Yes, exactly.
That reminded me, like, of reading old doctor notes in high school. I think doctors used to smell patients to like determine if they had diabetes or not.
Yeah, that's true. In the old days, yeah.
Those were wild times. Can't imagine doing that.
Yeah, that is definitely true. Okay. Okay. Oh, sure. Oh, we were talking about the volume. Okay. Right now, this is a picture of urethra. Urethra is responsible for transporting urine from the bladder to outside of the body. A is for a female, B is for a male. So when we think about the transport of this, the volume of it plays a huge part. If we're peeing frequently in a day, so as I said earlier, 1 to 2 liters a day is normal. If we have greater than those 2 liters, 2.5 liters or more, it's called polyuria. And that's basically a sign of diabetes, excess alcohol, caffeine, or kidney disease. It could also be a result of drug use. It could be a result of a diuretic, sickle cell anemia, or just you drank way too much water. You're drinking too much water, right?
Mm.
Now, if your volume is low, to about, like, say 500 to 300 to 500 milliliters of urine. So that's like 0.3 to 0.5 liters. That basically means you're dehydrated. Either that, you're losing blood, you have diarrhea, you have cardiogenic shock, kidney disease, an enlarged prostate. Now, if you have no urine or very little urine, that's basically a sign of kidney failure. So if you're not peeing, that's a problem, right? And that could be a result of obstruction. So something's blocking it. It could be like a kidney stone or a tumor that's blocking, or it could be a enlarged prostate. So again, by just looking at the volume alone, we can kind of have an idea that, hey, what's going on, you know?
Mm-hmm.
So, questions so far?
If it gets blocked, does that mean it goes it goes out the other end?
Not necessarily, no. It usually means that it's being retained. So you're retaining all that liquid. That's not good, right? Because
Like,
you're trying to get rid of some of that waste that you're generating.
can that cause like kidney bursts or something like that?
Yeah, if it happens too long, it will cause major kidney damage. You know, burst would be, yes, part of it, but other things will happen before that happens.
Okay.
You know, because then what happens? You're producing a toxic environment because that filtration. So like your kidney is going to be serving as a filtration system, and so you're going to have blood flowing through there. They get cleaned out, and it's going to be going back through that system. But if you're not filtering out anything, if you're not peeing, then you're not getting that filtration happening. And so you're just recycling and it's just building up on itself. So
That makes sense. It's like a vent filter.
yeah. And let's see what else. Yeah. Also, what kind of regulates that or allows that water to be reabsorbed or to be released is going to be your channels, your water channels, or your aquaporins. So if you don't have enough of those, then that can also lead to not being able to pee. So. No, actually, if you have too many of those, if you don't have enough, then that leads to you being able to pee it all out. Okay, let's move forward. Okay, so this here is so we know that blood is going to be flowing in the kidney, and so this is just a diagram of the kidney, and it shows you all of the different arteries that'll be entering and arteries and veins that'll be entering the kidney regions, right? Okay. I should say, let me take a step back. Okay, so if you take a look at this, so this guy is a nephron, and it's going to be like the functioning unit of the kidney. So it's going to basically responsible for cleaning and balancing out components. So what's going to be happening here? It's going to be recycling parts of the components, and it's going to be putting things back into the bloodstream. Okay, certain things are going to clean it up and then put other things back in. Okay, and we're going to talk about that a little bit later. Okay, so one of the things that are going to be happening. So the kidney is actually going to release an enzyme called retin into the blood. Retin's job is to basically process. It's going to convert angiotensin angiotensin, and the liver is actually going to release the angiotensinogen into the bloodstream. The retin is going to cut it, and so angiotensinogen is going to be a pre-pro enzyme. So then it's going to cut it to angiotensin 1. Angiotensin 1 is a proenzyme. So a pre-proenzyme is inactive entirely. A proenzyme, it is partially inactive. A lot of times that that region that keeps it from being completely active or keeps it from being inactive is often a transport signal. And so it gets to a certain point and then it gets clipped again. But in this case, it actually goes into the bloodstream, and then we get another enzyme called the ACE enzyme, That is a lung enzyme, and that's actually secreted into the bloodstream. And it actually cuts the angiotensin 1 to make it angiotensin 2. Angiotensin is the active form of the enzyme. So that enzyme stimulates the adrenal cortex to secrete aldosterone, and then aldosterone basically stimulates the uptake of sodium. Okay, so then now we're kind of tying that whole concept of kidney function into sodium uptake. Okay, and so we can start to see how it ties into what we say, our blood pressure. Right? Okay, let's see. So we also get, we're also going to get ADH is going to cause your aquaporins to move to start collecting ducts to the collecting ducts of the plasma membrane, and so that you're going to increase water being reabsorbed. So we're going to allow water to sit there and be more reabsorbed in the nephron of the kidneys. Okay, so, oh, sorry, this is Aquaporin. This is Aquaporin Channel, and so the way it works is that go back. So we get the uptake of the sodium ion. That uptake of the sodium ion is going to cause this channel to kind of open up so that we're able to sit there and allow water to move through.
That's how water gets filtered?
Yep. So when we think about our blood pressure is going to be a combination of 3 different types of pressure. So we're going to be looking at what we call our NFP, or the net filtration pressure. It's going to be the sum of the osmotic and the hydrostatic pressures. So we have the blood that's going to be flowing into the nephron. That's going to be a result of a hydrostatic pressure. In this case, it's going to be roughly, in a normal circumstance, about 55 millimeters of mercury. We have the capsular hydrostatic pressure, which is going to be about 15 millimeters of mercury, actually flowing out of the nephron. And then we have the blood colloidal osmotic pressure. I'm sorry, the blood colloid osmotic pressure flowing outside of the nephron. So overall, if we look at the total pressure, we're going to have a flow inward. Okay, so your net pressure is going to be heading inward. Okay, so during that process, as it's flowing in this earlier region of the nephron, you're actually going to get absorption of your amino acids. You're going to get absorption or reabsorption of your glucose, amino acids, proteins, vitamins, lactate, urea, uric acid, your sodium ion, your potassium ion, your calcium ion, your magnesium ion, your chloride ion, your bicarbonate ion, and water. So, and then as we move down, a little bit further, that's the things that have been reabsorbed. Some of those things are going to be recollected, and then they're going to be secreted into the nephron. So then you're going to get that urea coming back, that uric acid. You're going to also get some creatine, you get some drugs. You're going to get the hydrogen ion, and you're going to get the ammonium ion. As we go down further, we're going to absorb water. Then we're going to secrete urea. Then we're going to absorb those the sodium ion and the potassium ion and chlorine ion. Up here, we're going to get more absorption. Here, we're going to get some secretion. And then the last part, we're going to get absorption of water and urea. Okay. So it's just going to be a process of, if you look at it, it's going to be collecting. It's a nice way of recollecting all the things that you need. It goes through that filtration process where you're going to absorb some of the things, and then you're going to get rid of some of the things, right? So if we did an analysis of urine itself, and you look at the absorption and the absorption rates, I mean, what's absorbed in 24 hours and what's released in 24 hours, we get an idea of how much is it going to actually be filtrated, right? So like, for instance, at the end of the day, the total amount of water that's filtrated is 180 liters. The total of water that you're going to find in urine is going to be 1 to 2 liters, right? So we're absorbing the remaining part. So that's 178, 179 liters that is being reabsorbed by this process. So it does a really good job of filtering. Proteins, right? Full proteins. And if you actually take a look, that's one of the first things that gets filtered out up top or reabsorbed. We have no proteins. If you're a normal individual, you should have no proteins in your urine. That means that you're recycling all the proteins that are being put in your body. Okay, at least you're not getting rid of it through urine. You may get rid of some of it through defecation, but that's it. Chlorine, the chlorine ion, it's there's about 630 grams of chlorine ion that is being filtered, and you're only losing 5 grams in urine. So you kind of get an idea of how efficient your kidneys are functioning.
That's really high efficiency.
Yeah, it is. It's incredible what our body can do. Okay, so here, this is basically just showing you that reabsorption and secretion. And so this here is the lumen of the proximal tubule. This is the proximal convoluted tubule cell. And so if we take a look at this, right, so this process basically means that it's going to be a channel that needs energy to sit there and make it happen. So the ATP that drives this, it allows in the lumen, sodium to leave the lumen and go into the tubule cell. And then at the same time that's happening, you're getting the hydrogen ion released, or a proton released. And then that's similar here. So you have both your chlorine ion and your sodium ion. Here you have your calcium ion and amino acids. And so you're able to sit there and reabsorb those amino acids into that cell, right? And so then what happens is that these guys then get transported to the bloodstream, back to the bloodstream, and then they go through that process again. Are you with me?
Yes.
Okay. So if we look at the reabsorption of bicarbonate, so in the lumen, you're going to basically have bicarbonate, but you're getting that hydrogen ion being secreted back into the lumen. And so that bicarbonate is going to interact with the hydrogen ion, and it's going to form carbonic acid. Carbonic acid is an unstable acid, and although it happens pretty fast in real life, it separates and makes makes water and carbon dioxide. We also have an enzyme in our body, carbonic anhydrolase enzyme, which basically removes that water and makes carbon dioxide, but it makes it happen in a more efficient and controlled manner in our body. So we know that enzymes just speed up the reaction. They're just speeding up this reaction here. So then what happens is that we get water, and then we get carbon dioxide absorbed by the tubule cell here. That tubule cell, that carbon dioxide that gets absorbed, is then it reacts with the water, and then it remakes that carbonic acid, and then that carbonic acid makes the H+ ion and the bicarbonate. And then the bicarbonate is going to be secreted back into the bloodstream.
Just recycling.
Yes. Very, very efficient recycling. So in the case, we're never really losing anything. And we have this nice recycling system that we know that we need the bicarbonate to kind of help neutralize the acids in our stomach. And so they're going to be delivered to where they need to be so that they can do their job. So we know that that comes from the pancreas. So somehow the pancreas either generates or it collects the bicarbonate molecules that are needed for the neutralization.
Oh, okay.
Pretty cool. At least I think it is. Oh, by the way, you know that this is our last lecture. Did I tell you that?
Yep, Jesse told me that.
Excitingly said that, huh?
No comment.
No comment. Okay. So this basically just kind of tells you that you have this counter current going on. So you have water going in one direction, you have urea going in the other. Water going in one direction, going in the other. And so basically, at the end of the day, your urea is going to be collected. That's, you think about what's being released in terms of urea. Urea is one of the largest components released. But even in the case of urea, you know, we have 53 grams that is filtered. Of that 53 grams, we get rid of about half, which is about 25. We get rid of about 25 grams, and then we keep 28 grams. Okay. Okay, let's see. Okay, so hormones basically are also going to be influencing our blood flow and our glomerulus filtration rate. Or GFR. GFR and our renal blood flow. Okay. I need to fix that down there. So in the case of our sympathetic nerve, so epinephrine and norepinephrine, they're going to stimulate the extracellular fluid volume. It's going to decrease. And then you're also going to get, as a result of the epinephrine, the extracellular fluid volume decreasing. We're going to also get a decrease of our GFR, and then also of our our RBF. And so various hormones are going to play that part. The insulin is going to have that effect as well too. It's going to actually decrease the ECF, but you're going to get an increase of the angiotensin, and then you're going to get bradykinin, and your pressure due to your stretch there. And so as those things go up, as those things go up, they're also going to have an effect on the GFR and the RBF as well. I should probably point to that right here. So, and under those circumstances, we're going to get constriction, right? So we're going to get more vascular constriction. So under that case, in the case of prostaglandin, we're going to get, even though we're going to get a decrease in our ECFV, we're going to have no change or little change to the GFR, but the RBF is going to go up. And so we're going to actually have an increase in our shear stress in that situation. We're also going to get an increase in the angiotensin too. So in the case of nitric oxide, it's going to basically cause the stimulation of histamines, bradykinin, ATP, adenosine, and acetylcholine. Both of those are all going to end up causing the GFR to increase and the RBF also to increase. So bradykinin, which causes a decrease in prostaglandins and the ACE, will also cause an increase in those flow rates and also that filtration. And then natriuretics, peptides. So those are they're basically peptides that are going to stimulate the kidney to excrete sodium, so to kind of get rid of sodium. And so they also are going to cause the ECF to increase. They're going to cause the GF, the GFR to increase, but it's not going to have an effect on the flow rate. Okay. And so, do you know what the GFR? You know, it's you know what I mean by that? What's happening in that case as it increases?
Backs up.
It's what?
Backing up.
No, it's not necessarily that it's backing up. So if it's increasing, that means the filtration rate is increasing. That means that you're actually clearing out more components. It's not necessarily your flow rate is going to be increasing. It's not like you're getting more blood through. It's that you're more effective. So that means that the things that are responsible, those channels that are responsible for the absorption are abundant, right? So these guys are going to be stimulating those channels. So you have more of those channels at the surface so that you can take in more stuff. So your flow rate is not increasing. You're consistently getting the same amount of flow rate in this case, in this last case, but you have higher production of those channels to allow that to happen.
So in the case on the first two rows, it means it's doing a worse job compared to the bottom rows for the GFR?
Well, so you're saying down here, up here, the first two rows?
Yeah.
Yeah. So in this case, yes, you're not filtering more, you're actually filtering less. So in the case of when you're worked up, your fight response is up, you're not going to be, your body isn't going to want to waste energy about filtering anything, right? It's wants to go. So it's like, okay, we're not going to filter, whatever comes out is going to come out. Tough, right? So like if you think about a dog who gets real scared, like, or gets really excited, a lot of times will pee on itself, right? So if we analyze that pee is going to have a lot of stuff in it because you're not going to be filtering, you're not going to be reabsorbing as much stuff as you would. So does that make sense? Because your body is saying, hey, we need to get out of here. We can't worry about trying to use all this energy on recycling all of this stuff. We need to worry about getting out. So we want to make sure we're using that energy for that. In that case.
That's simple.
Okay, so these are going to be your waste products. You're going to have ammonia, urea, and uric acid.
Yep.
This is the chemical structure for ammonia. It has NH3. This is the chemical structure for urea. Let's see, C, NH2. Okay. And then this is your chemical structure for uric acid. So that one is okay. Questions, concerns, Cash?
Simple enough.
Okay, so this is just basically same figure that we saw earlier. And the thing is that we look at this is going to be like a feedback loop, a negative feedback loop. So we're going to get the regulation of this system by the regulation of these components, right? So what happens is as we go through and process this enzyme, we make more and more of this product. Well, this product is going to be responsible for causing sodium channel to be released, and it's actually going to end up causing a shutdown of the production of these enzymes. All right. And so it's a negative feedback loop like we've seen before.
Most of the body consists of that type of loop then.
What was that?
It looks like most of the body utilizes negative instead of positive.
Yeah, there's a few. I mean, like childbirth, I mean, the contractions and stuff, that's going to be a positive feedback. There's a couple of positive ones, but you're right, a lot of them are negative. So like if you get too much of a certain thing, then it kind of regulates itself. It goes back and shuts itself off by another mechanism. Okay. So that's it. That's all I got, which is like 2 minutes before time. Huh.
Yeah.
Compared to 15 minutes over. Okay. Okay. So what was I going to say? I was going to say something.
Was it about labs or the exams?
There no, well, you only have the simulations. That's it. That's all you have left, but I think it was that. And once you finish that, you're good to go. And then you just have your exams.
All right, cool. Should I just film me taking the exam and then submit that instead of doing Zoom record?
Yeah, that's fine.
All right, I'll do that then.
Well, I don't know. It depends on if Proctorial is working or not. So then I'll have to shut off Proctorial. So maybe we should plan it to do it that way. Set up a time.
Oh yeah, can I get the
Code?
code ahead of time?
Yeah. I'll email you the code.
Let's see.
But yeah, that's fair. I'll let you record it. That's fine.
For the labs, it's all of them, or is there just like or is there what am I trying to say? Do I have to do all the labs, or is it just a certain amount?
Only the ones that we have listed. Even though we didn't go over the material in class. And how many? I think there was what, 3 or 4 left.
There was a good chunk left for EXO.
Well, I know we're not doing them all. You only have to do the ones that I had listed on Canvas. Because some of them we did other things instead. We did either a physical lab to kind of take its place, or we did.
Let's see if West Hills would load. Okay. The site crashed.
Yeah, wasn't it? I remember, well, 2 of them should have already been done. I think there's 2 left for this week. There's one for Monday, and there should have been one for Tuesday. Am I wrong?
Well, so there's the 13 lab. Oh, that's just it. Exercise 8.
Okay.
I don't see any other lab around here.
Then that's it.
All right. Oh, thank goodness. It's only 4 activities.
What was that?
There's only 4 activities for this lab.
Good.
I think everything should be smooth sailing. Just got to turn in the task correction, do the quiz, and then the final exam, and then everything should be good.
Yep. You got half a week left, so that's good.
Thank you for everything for this semester. Learned a
Yeah,
lot.
no problem. No problem. Thank you for being patient. I appreciate it.
Yeah, well,
Okay, so I'm gonna let you go, and I'll see you. I'll see you when I see you.
all right.
Okay.
Well,
Take care.
you too. See you, Jesse.
Bye, Alejandro.